Welcome back to our high-yield biochemistry series. Today, we're diving into glycolysis, one of the most fundamental and frequently tested pathways you'll encounter. The key to mastering glycolysis isn't just memorizing steps. It's about understanding the big picture. What are we starting with? What are we making? And most importantly, what tells the pathway to speed up or slow down? Need to know the rate-limiting enzyme and its regulators. Let's start with a quick overview. Glycolysis is an irreversible process happening in the cytoplasm. In a nutshell, it takes one molecule of glucose and breaks it down into two molecules of pyruvate. The net equation shows we start with glucose and end up with two pyruvates, two net ATP, and two NADH. Essentially, we're converting glucose into storable, usable products for other pathways, like the citric acid cycle. Now for the pathway itself. Glucose enters the cell. Say, after you've enjoyed a milkshake. First, it's converted to glucose 6-phosphate, then to fructose 6-phosphate, and then to fructose 6-stay. After a few more reactions, you get phosphoenol pyruvate, which finally becomes pyruvate, marking the end of glycolysis. Let's focus on the crucial enzymes. The very first step, converting glucose to glucose 6-phosphate, can be done by two different enzymes. Glucokinase, this is super high yield, so listen closely. Hexokinase has a high affinity for glucose, meaning it works constantly in all your tissues to maintain a baseline level of glucose metabolism. Think of it as the everyday workhorse. Glucokinase, on the other hand, has a low affinity. It's found mainly in the liver and pancreatic beta cells and acts as a glucose sensor. It only kicks into high gear when glucose levels are extremely high, like after a very carb-heavy meal. When hexokinase gets overwhelmed, glucokinase steps in to help manage the excess. So, hexokinase for basal metabolism, glucokinase for high glucose emergencies. Remember this difference. It's a favorite on exams. Next, we hit the most important enzyme of the whole pathway, the rate-limiting enzyme. This is phosphofructokinase 1, or PFK1, which converts fructose 6-phosphate to fructose 16-bisphosphate. Since it's the rate limiter, its regulation is critical. So, what controls PFK1? It's actually quite intuitive. Think about the goal of glycolysis, to make ATP and pyruvate, which can become citrate. So, if your cell already has plenty of ATP and citrate, does it need to run glycolysis? Nope. That's why ATP and citrate inhibit PFK1. They are feedback inhibitors. Conversely, what if your cell has a lot of AMP? That's a signal that you're low on energy, you're low on ATP. So AMP promotes PFK1, pushing glycolysis forward to generate more energy. Thors. Always think about the cell's energy status when you're trying to remember these regulators. Now for a little nuance. There's a side step here involving an enzyme called phosphofructokinase 2, or PFK2. PFK2 can convert some fructose 6-phosphate into fructose 26-bis. Why does this matter? Because fructose 26-bisphosphate is a powerful activator of our rate-limiting enzyme PFK1. It basically supercharges glycolysis. So, what controls PFK2? Hormones, insulin, which is released when blood sugar is high. Finally, we have the last step, converting phosphoenol pyruvate to pyruvate. This is catalyzed by pyruvate kinase. And just like PFK1, pyruvate kinase is inhibited by the products of the pathway ATP and citrate. Again, if you have enough energy and enough downstream products, you don't need to make more. The pathway shuts itself down, too. And that's really it for the high-yield points of glycolysis. You might be wondering about all the other enzymes and intermediates. Honestly, for most exams, you don't need to memorize every single step. Focus on what we've covered today, the key enzymes hexokinase, glucokinase, PFK1, and pyruvate kinase, especially their regulation. Know what promotes the pathway and what inhibits it. If you get stuck on an exam question, just ask yourself, what is the goal of glycolysis, and what would the cell want to do in this situation? 4. If this video helped you, please drop a like and a comment below. And consider checking out our Patreon to support the channel. Thanks for watching, and good luck with your studies.